All right, everyone, I want to thank you for coming today. I'm going to let Isaiah just introduce himself, but thank you for coming and have a wonderful spring break.
All right. Well, hi, my name is Isaiah Tolo, and I'm a PhD student here at the University of Minnesota. And my home research institution is the Minnesota Aquatic Invasive Species Research Center. And this project that I'm going to be talking about today is a really cool kind of blending of the two different worlds of science that I've worked in. I've always been interested in microbiology and also in conservation sciences. So this is kind of a cool blending, and it's a really truly unique application of the skills that I have so far. So that's why I'm interested in it. But I'm also really excited to see whether we can use these viruses that we've been uncovering in the wild as potential biocontrol pathogens for controlling aquatic invasive species. So kind of getting into the story, I guess this thing is going to be up here, but that's all right. It might block out something. I might have to explain what's in that corner at some point. But this is just sort of the story of where common carp has come from. So some of you may know common carp is a large cyprinid species that was introduced into the United States in the mid-1800s. And it was introduced because it was considered a desirable fish species to like kind of supplement people's diets and to have as a cool sport fishing fish, and no one really knew how it was going to take off. And now today it's really distributed quite broadly. It's totally naturalized, so it reproduces on its own without any help from us. And it has started to cause some really serious environmental consequences. So that's why we care about it. The mechanism of this is basically that it can cause this alternative stable state switch in shallow lakes and shallow rivers. The mechanism of that is basically that these carp are omnivores. They're really large bodied. They can reach these really big populations because of how fecund they are and how fast they grow. So when they're endless rooting around in sediments for their prey, and also interested in like seeds and other things that they can eat in the sediments, they disturb macrophytes, and they can cause macrophytes to decline, which can then take some of the pressure off of algae in the lake. So you can have this kind of algae-dominated system. They further contribute to that through their excretions of their populations, and also through bioturbation, as they stir up sediments and unbury nutrients from them. Those things can feed into this same kind of pathway of causing algae to be the most dominant in the system, and that can shift to this turbid state. And then that's when you start seeing some of these environmental issues. So decline in water quality, which is bad for native species, bad for carp too, but they can tolerate it, and their populations can continue to grow. If you add that to their kind of direct competition effects, then it's really sort of this really bad feedback loop for native species, where their prey items are being taken away, their habitats being taken away, and the water quality is bad. And then again, this isn't so bad for carp because they can handle that worse water quality, and they continue to increase even though native species are struggling. So this is just like a nice kind of bird's eye view of how these effects are playing out across the state. So basically what we're looking at here, this is a figure from Epidemics lab. It's really nice. I use it in a lot of presentations. But what we're looking at here is, I don't think, oh yeah, that was a fireworks, great. On this side, we're looking at a map that shows the density of carp as a proxy. The proxy is carp kilograms per net, and basically what we're looking at is when you see like a big pink dot, that's where carp are more dense on the map. And then on this side, you can see where plant richness is in Minnesota, and you can see that these really don't overlap very well with carp kind of pushing out plant richness in this part of the state. So there's a suite of different options for controlling carp, some that have been used for a long time, and I would kind of consider like the de facto way that maybe their carp are regulated by the DNR, for instance. And then there's a whole bunch of different methods that they're controlled by. I want to camp on harvesting for a second because I recently found this out and thought it was interesting. So this is a plot basically showing the amount of harvest of different commercially fished freshwater species, and that's in metric tons. And that corresponds to these bars. So the top two bars here, this is common carp bars, and these are buffalo. So these are kind of the two species of interest to a lot of commercial fishermen. And the black line is showing the basically the total value in millions of dollars for freshwater fisheries in the Mississippi River Basin. And as you can see, this black line has just been kind of tanking up until recently. And the reasons for this precipitous decline in the value of commercially fished freshwater species, we're not really sure why it might be, but it could be related to taking off of aquaculture. It's been a really fast-growing industry. Also, the discovery of contaminants, especially in some of these rough fish like carp, and then just changes in people's preference about what they're going to eat. And something that you can't see here because of this little thing here, but what I have up there is actually the value of common carp and how it's changed from the 1950s up to today. And basically, there's been, I mean, carp are basically worth something like 8 cents a kilogram or 8 cents a pound at this point. And buffalo have also decreased in value as well. And that's kind of a problem because the fishermen are out there really to get their money off those buffalo. And so now they're not getting as many permits to fish those places, and there's not as much fishing pressure on carp. So that's just kind of an interesting thing about commercial harvest, which I would consider sort of the de facto way that carp are being controlled. But there are these other really promising methods for controlling carp. Chemical control is sort of the way that you would just totally annihilate a population of fish. If you're just too far gone, you want to draw it down and get rid of all the fish. You could use barriers of deterrence to keep carp from recruiting back into those areas. You can also use removal methods. Some of the new ways that this is being done is actually using attractants to get the carp to come into one place, and then you can remove them more species specifically and with more effectiveness. And then also biocontrol using natural predators of carp eggs. So bluegill, places with marshes that have a lot of bluegill in them are generally not good places for carp to recruit back out of. So one more interesting method of control, actually I didn't mention genetic biocontrol strategies. Some of those are kind of on the horizon and are really promising as well. But pathogen biocontrol is interesting because it's sort of a tool that could be self-disseminating. So for a lot of things that would be harder to scale up to the scale of the problem of common carp, may not be a problem for pathogen biocontrol because it would kind of disseminate itself. I guess it depends where you're coming from, but you may find this to be kind of an alarming idea. Like, maybe you get this idea of like a host jump, and then there's all these terrible non-target effects. But to me, this actually seems like what could be more natural. Like you're using like this tiny little molecular predator of your aquatic invasive species that's perfectly adapted and honed to decrease populations of that species based on sort of this like density-dependent relationship. So the problem that we have is a density-dependent relationship with carp, where when they get to be too dense, then you start to see this stable state shift. And pathogens like herpes virus, for instance, are ones that they kind of thrive in those types of environments. So I think this is a really interesting and promising tool. And it actually has been proposed as a potential control method for carp in Australia. So there's nothing, there isn't like a control strategy that's already like funded or in the works at the moment, but there's a lot of research that's been dedicated to this in Australia. And the interesting thing about their system is that KHV has never been detected in Australia to this point. So that means that their population of carp is completely naive to the virus. And that's really beneficial because they're going to be likely more sensitive to the virus too. So the idea is that you would introduce the virus and then you'd have kind of this like cataclysmic knockdown of the population where just millions of fish die off. But now the interesting thing with KHV is that after that initial like large-scale mortality event where you have like the really obvious, fish floating to the surface, thousands of them stinking and getting on the news and everything like that. But after that, KHV continues to persist in those populations. So one good way to understand this is if you know anything about chickenpox. So I got chickenpox when I was a kid or a baby or whatever, I don't remember it. But in high school, I remember at like like had like this rash on my side and like hurt and just got worse and worse until I went to the emergency room because it felt like my ribs were breaking. And the doctor's like, you have it's VZV, and I was like, oh, what the hell is that? And it turned out that this is actually a recrudescence, or it's the virus coming back out of latency and causing these lesions again. So it's the same virus as chickenpox, and it can basically go into a subtype of your cells, neural cells in the case of humans, lymphoid cells in the case of Carp with KHV, and then it can be reactivated by different environmental stimuli. For Carp, it's a little bit more reliable, that reactivation. So basically, if the temperature is between 18 and 25 degrees Celsius, then you're going to see reactivation of the virus. So what's cool about that is that any fish that survive the initial outbreak, if the virus was transmitted to them, they can transmit the virus back to new naive populations. Those new naive populations are going to be like baby carp that are coming in from the marshes to join the adult carp in the river system or in the lake system. Or it could just be populations that were more like far removed and they just haven't been exposed to the virus yet. I put these little stars in here because with any pathogen biocontrol strategy that's been tried in the past, there's always been other management strategies that have been implemented at the same time. So whether that be more conventional methods like removal events or things like removal strategies or things like that, or whether it be serial release of additional pathogens, so a more virulent strain of the virus or a different virus that the population isn't already exposed to. And the reason for that is just that there's always kind of this arms race between pathogens and their hosts. So they're always kind of working to establish this kind of equilibrium where not all the host dies, and so the virus can continue to go on. So a couple of different examples of this. I don't know as much about this one, but viruses and entomopathogenic fungi have been used to control red ants in North America. These two are a little bit more famous. Marion Island is an interesting one where feral cats were actually completely eradicated from Marion Island using a combination of trapping, shooting, and feline panleukemia virus. So they introduced this virus and they were able to completely eradicate feral cats on that island. They weren't just killing them for fun. The feral cats were totally decimating like migrating seabird populations on that island. And then probably the most famous example is the European rabbit in Australia. So this was causing huge expensive effects across the entire continent wherever there was pasture land for sheep. And they used conventional methods like horn ripping and shooting and trapping, along with two different viruses, myxoma virus and RHDV, a rabbit hemorrhagic virus. And they also introduced a insect vector at one point as well. And though they didn't completely eradicate European rabbits, they're still there. Their population is like 1% of what it used to be previously. So those environmental effects have been pretty much eliminated, and they've saved billions of dollars. So where would we begin for pathogens in Minnesota, using pathogens for biocontrol in Minnesota? So obviously we want something that's safe, and that says effective right there. We want something that's safe and effective, but kind of unpacking that. Safe means that it's going to be species specific. It's not going to infect any other fish, only the aquatic invasive species of choice, and that it's going to have a slow mutation rate. So I'll unpack that one too. Some viruses like the flu, it's an RNA virus, and it likes to jump hosts. It's kind of like its life strategy is if I can take on mutations, I'll be really flexible, and then I can be in a pig today, and I can be in a chicken in a couple of years, or whatever. But for some of these DNA viruses, they're more like us. They don't really want to change. They will take on a few mutations at a time, but they're not going to be jumping hosts anytime soon. In fact, it can take thousands or millions of years for a DNA virus to jump hosts. As far as effectiveness, first of all, we want something that's just has good disease dynamics for pathogen biocontrol. So kind of a balance between being able to be transmitted readily and killing off a good percentage of the population. Also, we would want to think about, and when we think about effectiveness, we also want to think about like our ecology here in Minnesota. So we need these populations that we're going to be targeting to be dense, which Fortunately or unfortunately for us, they are. And we also want them to be naive and susceptible. And again, I kind of want to touch on this because this isn't like completely clear, but you have to think about a population being naive and susceptible over space and time. So not just like thinking about it like a map where you have positive lakes and negative lakes, but also thinking about how carp are going to recruit into those lakes over time. So you'll have new susceptibles, or there can be turnover and you'll have new susceptibles that way. So we're approaching this from a very conservative approach where we're going to try to use something that's already here. So this isn't really like the classic way of using pathogens for biocontrol. We usually use an exotic pathogen. And it's a little bit of a scary situation because you have no idea really. I mean, you might have some idea from looking at other regions in their ecology, but you haven't actually seen the pathogen interact with your system. So this is a good way of making sure that you can actually have study sites at your fingertips. You can actually see what effects it's already having, and then you can decide whether to proceed or not using that information. So kind of our strategy of going about this is sample fish from fish kills of native species in carp. So basically, a fish kill is if you go out to a lake and you see a bunch of dead fish in one spot. That's a fish kill. If it's 5 fish, it's a fish kill. If it's 1 million fish, it's a fish kill. But we wanted to look for things that were in native species so that we can rule those things out if it causes disease in native species. And we also wanted to find things that were in carp so that we could consider those as candidates. But if we found those same pathogens in healthy populations, then it could just be a commensal microbe, or it could be just a highly opportunistic pathogen that is going to only cause disease once in 1 million times. So using that as kind of our filter, we don't want things that make sick, that make native fish sick, and we don't want things that are found in healthy carp. We just want things that are found in sick carp. And then we could move that on into doing like a disease trial with those candidate pathogens, basically trying out a couple of native species in our carp and making sure that it's as virulent as we want it to be in carp and that there's no replication or disease production in native fish. So thinking about that sampling strategy, we used a couple different ways to identify these pathogens. We used a polymerase chain reaction, which is useful for if you're looking for something specific. So if you have like a panel of bacteria or viruses that you're interested in, you can use that to amplify their DNA out of a sample and see if they're there. NGS is more kind of like this, like a catch-all in this situation, where you can use it for viral discovery for things even if you don't know are there. So it's more kind of like a broad net. And then cell culture is kind of like the gold standard. If you can get a pathogen, or cell culture, or just culturing pathogen in general, if you can get it to grow and you can isolate, get a nice isolate of it, then you can actually move on to those disease trial steps. So again, for fish kills, we used a lot of different methods, but what we found out through the course of the study is that getting good samples from fish kills is actually really difficult. A lot of times you're going to get them, and for lack of a better word, they're already rotting. So those tissues aren't going to be good for histology, and rarely are they going to be good for parasitology, bacteriology, or virology. These pathogens kind of require a living host because that's what they're using to replicate. That's their system that they live in. And so once that host is dead, you're a lot less likely to be able to get those pathogens. But you can always find their DNA using PCR or using NGS. We also screened healthy carp. We used PCR for a couple of like specific pathogens. So we looked for carpedema virus, string viremia carp virus, and koi herpes virus. I already explained why we were interested in this one, but these two are also pathogens of significance for common carp that are species specific or have a level of species specificity. So we sampled a lot of healthy common carp, like 600 fish. We sampled fish kills from 30 lakes. This is just a list of different native species that we were able to sample too. And the cool thing about this is that even though we're looking for this pathogen biocontrol agent, we're also able to use this study to kind of learn more about fish kills in Minnesota and to kind of increase the capacity of the DNR and our lab to investigate those, which could be useful for conservation of those species as well. So I want to highlight a couple of interesting outbreaks of of disease in common carp. The first one being KHV at Lake Gillespie in 2018. So this wasn't the first time that we detected KHV in Minnesota, but it was a example of one we were able to get a little bit more information from a typical mortality event. So you can see we're just doing transects, driving along the lake in a boat and counting dead fish as we can see them. But it kind of gives you an idea of what we're actually looking at when we go to one of these fish kills. This is actually a good example where we have a boat and not just our legs. But this is kind of what you'll see is just around the whole entire perimeter of the lake, or maybe all on one side of the lake, you just see all these floating corpses. But these are all pretty large fish, and they're just the ones that are floating. So there's a lot of things that you're missing. KHV has these really characteristic like hemorrhages on the skin, loss of scales. This is kind of the most characteristic thing. It's a little hard to see, but this gill is just super chewed up. So these lesions on the gill are something that is common during KHV infections. Another interesting mortality event was a co-infection of KHV and CEV on Lake Current in 2018. So this is interesting. This co-infection is interesting for two different reasons. One, because CEV had never been detected before in the USA, and also because co-infections had never been described anywhere in the world. So this was a really interesting and novel finding that we were able to pull out of this. And then this is kind of a breakdown of some of the PCR results that we have, just so you can get an idea of what we're looking at. But basically, we were able to find KHV in a whole bunch of common carp only mortality events, and some of those had co-infections with CEV. We also looked at SVCV because CEV and SVCV have been found in co-infections previously, so we tested all these samples as well, but they were all negative. And then our survey lakes were all negative. You'll just have to trust me with that part. And also our fish kill lakes were negative. So that's nice. We kind of made it through that first filter of finding candidate pathogens. And we were able to find both of these pathogens. So just for your interest, this is a video of a co-infection in a fish. This fish was actually collected from a lake which had previously had an outbreak of KHV, and we went back to sample and just see if we could detect the pathogen again. Just poking him there so you can get a better view. It's kind of sad and gross, but also really interesting. So speaking of samples from lakes that had infections previously, these are examples of a lake that we sampled for an outbreak event that happened in 2017, and then we went back and sampled it in the summer, kind of the heat of the summer, to see if we could find KHV again. And we found KHV in 3 of 8 of those samples, and one of those samples also had a co-infection. So this is a really small sample, but assuming that it was random, which some actually felt pretty random, didn't it? We were all over that lake. But assuming this is a random sample, that would mean that there's definitely, those viruses are still distributed in those lakes even a year after mortality event occurs. And I just got some little videos here so that you can kind of see what this looks like. Never been electrofishing. It's pretty frustrating. But you basically put a current in the water, it stuns the fish, and you're able to kind of pick them up and toss them in the boat. So the reason why we sampled in the summer is because of those temperatures that are like permissive for KHV reactivation. But I also was curious about sampling in the wintertime. So we sampled Lake Tatanka, which had an outbreak in 2017, but we resampled it in the winter just a few months ago. And in that case, we sampled a lot more fish, but only 3 of them were positive for KHV. And all of those were blood samples, which makes sense because the virus actually goes into latency in a lymphoid cell that's found in the peripheral blood. These were collected using a winter seine, so this is kind of a choppy video, but basically we're out on the ice, and these guys cut holes in the ice and drop nets down, and they're able to pull up buffalo and carp, and it's one of the ways that they make a living. So this is sort of the distribution of KHV and CEV during our study. So what we're looking at here is black dots are where KHV occurred by itself. Red dots without a bullseye in them are CEV by itself, and red dots with a bullseye are co-infections. The diamonds are places where we weren't able to those are places where we weren't able to get samples from. And what's interesting about this is just that you kind of see these two kind of clusters. This one is in the this one's in the Rock River system in Wisconsin, and you can see that there was co-infection, and then we also detected KHV by itself in two of those. And then this is all a bunch of lakes that are kind of connected to the Cannon River and its watersheds. And these occur over 2017 and 2018. And we don't know for sure that this is like the actual distribution of KHV and CEV in Minnesota. There could be some sampling bias because we made friends with these guys, and I emailed Dan a lot to get those samples. So that could be a reason why we found those clusters. So more intense sampling is going to be a way to kind of fill in this map or see what the actual distribution is. But at any rate, this distribution is probably not from an introduction of KHV in 2017. It probably happened earlier than that. This map is showing kind of the same thing. The black triangles are KHV by itself. The empty triangles are CEV, and then the ones with dots are the co-infections. And these are overlaid over carp density data in the state. So basically, this is average catch per gillnet, and the bigger the circle is, the more fish were caught in those average gillnets, and that's over 20 years. And just kind of a visual thing here is that it almost looks like some of these outbreaks are occurring where carp are more dense, and that might seem kind of like a no-duh, like the carp are there, and so the virus is going to be there. But it will be interesting to see if this pattern kind of continues as we keep sampling. And then looking at this kind of like on a kind of continental level, there are other outbreaks in California and Texas that I don't have up on here. I just wanted to focus in on kind of where most of the outbreaks have happened in the USA. The first outbreak of KHV was actually detected in New York, but it was in ornamental koi at a koi show. So this is kind of an interesting thing. The hypothesis is that KHV is actually spread worldwide through these koi shows, because people from all around the world will come with their beautiful koi, and they'll put them all in like a couple of tanks together, and they'll all commingle, and then they'll take their fish back home with them and go back to their home countries. So right after that outbreak, a few years later, there was an outbreak in a wild population of carp. And so this kind of led to the idea that the way that KHV has been introduced into wild carp populations is through accidental or intentional release of koi. And then since then, there's been a lot of detections along this kind of USA-Canada border, with a few kind of detections in the Great Lakes. This is actually not in the lake, it's actually in a lake close to the Lake Michigan. And then we're sort of the easternmost cluster of events since the initial detection. So in addition to just finding these things on the map, we also wanted to look at their genetic information and see if we could find out more about them that way. So this plot is basically showing percent identity of our genome that we were able to sequence from one sample, compared with all the other genomes of or some of the other genomes of KHV that we could find in the NCBI database. And basically what you're looking at is percent identity on this matrix. So with it going basically from 100% to things that are not 100%, both being more blue. What's interesting about this is that KHV is sort of separated into these two different genotypes, European and Asian genotypes. And we found that our strain kind of clustered together with the European genotypes. Just kind of a caveat here, though, it's kind of a weird thing about KHV, is that the low end of the scale is 99% identity. So there's really not that many mutations that you can use to tell how these strains are phylogenetically related to each other, but there is enough to distinguish these two different genotypes. And kind of another way to look at this, it's a little frustrating, this is covering this, but you can look at just an individual gene that has some distinct markers in it, such as insertions or deletions or single nucleotide changes. And you can just sequence one gene. And if you can do that in like a tissue sample, for instance, you're going to be able to get more strains, maybe where you can only get one from a really quality tissue. So in the red box, these are all the strains that we were able to get, we were able to amplify the thymidine kinase gene from. And this is an alignment of all all the multivariate sites in that gene. So it's 1,000 base pairs long. I just picked out all the places where it's different among different strains. You can't see it, unfortunately. You can see this little change here, and you can almost see the deletion that happens here. But basically, there are some mutations that differentiate some of the strains that we found in our study, which can mean that there's a few different strains that are circulating in the in wild carp. Another interesting thing you probably looked at right away is this strain that's typing as the Asian type strain. Blue is Asian type and yellow is European type. Sorry for not saying that. But this was our one and only isolate that came from an ornamental koi in Minnesota. So this was kind of interesting. This guy had it at a koi show and it died, killed a bunch of his other fish too. We were able to get tissue from that, and it turned out to be from the Asian type. So this doesn't necessarily like debunk that KHV was introduced through release of a koi, but it is kind of an interesting thing to think about. In the future, maybe through more relationship with people who are hobbyists in the koi trade, maybe we can start to look at some of the strains that cause disease in Minnesota and the koi community, and maybe see if there's any overlap of strains between wild and ornamental koi in Minnesota. And kind of going further with this is looking at the genotype of CEV. So CEV is a little bit easier to genotype because you can use a small fragment like this one, and there's actually mutations there that you can build a phylogeny off of. Similar to KHV, CEV is like divided into these different large like genogroups. There are 3 of them: genogroup 1, genogroup 2B, and 2A. And what's interesting here, these are our strains that we were able to get sequences from, and they're all clustering together in genogroup 1. And genogroup 1 only infects common carp, so it doesn't infect any koi. No one's ever detected that. Genogroups 2, A and B can infect carp or koi. But this is even a little bit more strength. This puts a little bit more evidence that there might be other methods of, there might be another etiology of KHV in Minnesota and wild carp other than just release of ornamental koi. Maybe it's going to be interesting to figure out what that might be. So, do we have 2 options for biocontrol agents for common carp biocontrol in Minnesota? Kind of yes and no. So they do, both of these pathogens, Carp virus and KHV, both kind of satisfy these traits that we wanted, where they're species specific, high mortality, easily transmissible. They're both DNA viruses. But KHV has just been better studied because of its threat to global food production when it comes to raising common carp in places where common carp are raised for food. And also because of its threat to the koi industry, which may seem kind of trivial, but some of those fish are worth like millions of dollars each. And so the person who bought that fish, this is a scary idea. But also it's been studied in relation to its potential value as a biocontrol agent for common carp. So because of that, there's just a little bit more information. Also, one of the big kind of like dings on CEV is that there are no culture methods for it yet. So no one's actually figured out how to grow this in cell culture. That pretty much means that it'd be difficult to do any kind of cell culture or to do any kind of disease trial with it. So could KHV work in Minnesota? So there are some lakes that are already affected by KHV. So does that mean that we can't use KHV anymore? This is kind of something I've been like trying to think about. It's like, well, they're not naive now, but we didn't really expect them to be. We were looking for something that was already there. So that kind of goes back to thinking about naive populations as being spread out in space and time. So what we want to know now is what kind of impacts does KHV have in lakes that are already affected? Like, does it continue to cause mortality, or is it just this one big one-off event and then everything else is kind of trivial and the population recovers? Or does transmission still occur, and does mortality continue after initial outbreaks? Specifically, we'd be interested in whether mortality is occurring in recruits of carp, so to see if KHV biocontrol could control that recruitment. So we don't have a lot of information on this, unfortunately. That's why I showed you that video, so you can see how hard my job was. But if we were to I'm taking like all the different estimates of mortality that we were given, kind of generalizing them into these bins of 50 to 100 fish, or 100 to 1,000, 1,000 to 10,000 fish. The minimum that we had in any one spot was 50 mortalities, and up to 10,000 mortalities. But the average of those, you know, it may seem like kind of like a lot of fish, especially over 20 lakes. But considering that some of these carp populations can be, you know, the tens of thousands, it's maybe not that striking of a mortality rate. We also did some transects of mortality on Lake Gillespie, where we basically drove the boat around for half-mile shoreline transects and counted everything that was floating or washed up on the beach. An average of 4 transects is 160 fish. So if you extrapolate that over the 14 miles of shoreline, you get this, you know, 4,500 mortality rate. So kind of a caveat for this, though, is that counting mortalities of fish is really difficult if you're just trying to do it by eyesight. You're going to be, you know, you're going to be somewhere, the whole duration of the outbreak has been occurring and you haven't known about it. Then you hear about it and you go and investigate the kill, and you may be here, and fish that died here may already have been degraded and they may be gone, or scavengers may have taken them. Additionally, when you get there, things that sunk to the bottom are things that are just obscured by a little bit of turbid water, or that are under a log or something. You're just not going to be able to count them. Additionally, this is just kind of a sizes of carp that we found in mortality events. And one thing that has been kind of annoying for me, because I'm really curious about this possibility of KHB affecting recruitment, is we didn't see any of these young of the year carp in these mortality events. So young of the year carp would be something like 9 centimeters. These are all like 25 centimeters in out. Which isn't unexpected. It can be really difficult to find these young of the year fish, but it would have been really nice to be able to see if they were kind of included in some of these mortality events. And the reason why, again, why we're interested in that is because carp have sort of like this source sink method of recruitment. So this isn't a lake that actually has KHB in it. It's just a lake near my house. But if this lake had KHB in it, the adults would be hanging out in the main lake, and they'd be using that as like a feeding ground, kind of a staging ground. And then in the spring, they would move to potential recruitment sites. If the water is high up enough and they can get through a culvert or something like that, they'll go into these marshes and spawn in there. And then at some point, those young fish are going to move back into the main population, and that's when they could get exposed to KHB again. So really, these are the guys we would be targeting with the biocontrol strategy, and these are the ones that would be kind of acting as the reservoirs. So that kind of begs the question, like, does KHB affect young carp? So there's been 2 studies that I put up on here. This Perelberg study basically looked at really small fish, 2.5 and 6 grams, and looked at their mortality rate compared to larger fish. And it looks like there was higher mortality compared to the larger fish. And then this study is looking at, it's more of an environmental study. And this is the number of individuals that were caught in their survey. And these are the standard length sizes. And positive or negative for KHB are indicated by these white for negative and gray for positive scale bars. And what's interesting that you can see here is that these younger fish, basically none of them have the virus. So, but you see in the older fish, and this is only a year and a half after or 2 years after the outbreak. So it's not like it was 10 years ago, and these fish aren't like shedding or something like that. They are shedding the virus. But these larger fish seem to have more prevalence of the virus, which means that they got hit with the virus, and some of them survived. While these fish, maybe when they get the virus, they just die. And so you're not able to detect many of them that have the virus. They're still alive. So some questions and next steps. I'm interested in whether KHB mortality, well, I know that these are underestimates, but I'd really like to know what those actual rates are. So kind of the ideal method of going about this would be to do some epidemiological modeling. We need some data to fill in that model. So being able to estimate transmission and mortality rates from local strains of carp and local strains of KHB would be important. And then also just doing a better job surveying these mortality events. Now that we've like honed in on this virus, we can kind of focus on it and put our resources into that and get more information. I'm also just interested in whether KHV causes high mortality in young fish. So does it cause mortality in recruits? And you can do that in the lab with disease trials, similar to those other studies we were just looking at, and also survey exposed populations to see if we can find those recruits and see if they have prevalence for the virus. Here's just a little video of a baby fish that I helped raise in the Epidemics lab. So basically the major challenge that we're facing in this study is being able to culture these viruses. So as I mentioned before, we can't culture CEV. There aren't any methods out there for doing it. We'd have to be the ones to discover it, which Somesh will do and be rich and famous someday, but right now we can't do that. And it's also very difficult to culture KHV. The reason for that is likely due to just the kind of a mismatch of our survey design and what you need in order to culture KHV. So I just kind of threw this up here. This isn't like the best sample that we had, but a lot of our samples look like this. And that's a big problem for culturing viruses because that virus isn't like it isn't like a little living entity of itself, like a bacteria with its own cellular machinery. It's like a USB drive. And in order to work, it's got to be plugged into a living host. So as soon as that host dies, you may really start to lose your chances of being able to culture the virus. So we tried culturing using frozen versions of these things on a bunch of different cell lines, and we just haven't been able to culture it yet. Also, we tried to use some of those frozen stocks to infect live fish in the lab. Tried to just go ahead with the disease trial, but again, there just might not be any viable virus in those samples. So a way around this that we're going to do, and it's going to be great, is that we're going to screen KHV carriers from previously exposed lakes. So one of the really cool advantages of studying KHV is that it sticks around in the environment. And we've even been able to go back to a lake where an outbreak occurred a year ago, and we can find KHV again. So if we screen some of these carriers from the wild, pick out some of the positive ones, and bring them back to the lab and house them for a while, we can house them at those permissive temperatures where the virus is reactivated, or we can stress them a little bit to get the virus to reactivate. And then we can use their tissues to start our virus isolation in stocks. Additionally, I hear that these are going to be very hard to manage. So we can cohabitate some of those infected reservoir fish with more manageably sized disease-free carp and lower their temperature or raise their temperature outside of the permissive range of KHV. And we can create basically these latent carriers that we can have in the lab and have a lot more opportunities to do this viral isolation. So ongoing research, viral isolation, we're going to do it. It's going to work. Moving on from there, we'll want to characterize the isolates that we find using in vitro and in vivo disease trials. So we can learn more about like whether they're seem very virulent or whether there's some that aren't as virulent as others, and we can kind of pick out particular candidate strains that we can move on to do genome sequencing with, which will let us kind of look at the molecular genetic underpinnings for why certain strains are more virulent than others. We'll also continue surveys. So I think there'll be more outbreaks in 2019. There were more outbreaks in 2018 than in 2017. And that may be because of reporting, or maybe because, you know, the virus is still just kind of blowing through some of these populations. But that'll give us opportunities to get to kind of put new points on the map, and also opportunities to just go out, like, again, we have those study sites at our fingertips, and we can learn about what the virus is doing when these outbreaks occur. And we'll, you know, focus in on looking on some of these population impacts, specifically looking at whether we can impact recruitment with this strategy. And then we'll also be doing some infection trials as well. And things that are kind of later on the docket are disease modeling. So using some of this data, we'll be able to kind of refine a good epidemiological model, which will answer a lot of questions for us, let us make some predictions. And then even further on is risk assessments, and then regulatory and public consultation, which is something that we're already doing now. We really want to socialize this idea, because it is kind of a either perceived or in reality is a kind of a high risk, high reward type of tool. So how can you guys help? I wish that there was like thousands of people here right now so that I could be like, go on, if you see a fish kill, especially if it's, well, whether it's native fish or if it's common carp, first of all, call the duty officer at the DNR and be like, I saw this fish kill, and then they'll investigate it. And then right after you do that, just jump on your smartphone and go to this website, z.umn.edu/fishkill, and you can just put a point down. You can take some pictures. You can put some information down and like give us like an idea of what happened at that fish kill. And the second that you send that, it'll pop up on Nick's desk, and he'll go and investigate it. Just kidding. I'll go investigate it. And this is just a great way to like kind of continue populating this map so we can learn more about how fish kills just even affect native fish. But it's also a great way to keep this this study going. And with that, I'd like to acknowledge all the great people who worked on this project. Somesh is always on the ground in the lab working on this, and Nick is my PI. And all these people have really made this possible. The DNR Path Lab has been really great, responding to some of these kills and getting samples for us. So it's been really nice to work with them. And also, yeah, just a thank you to the Bajer Lab for lots of help over this. It's like little helps over a long period of time is this big thing. And then also, thank you to our funding agencies. And with that, I'll take any questions.
What kind of virulence, you might not know the answer to this, but can you speculate as like to the level of virulence that would make it possible, feasible, economically viable to use this? And what metric is that, like a rate of transmission or like a mortality rate? Or
so I think you already know the answer to this. It's so like if you were to calculate like a basic reproductive number, that would be a good way of just kind of like a basic idea of like in ideal circumstances, is this going to cause an epidemic or not? Sure. So that's something that we can get from doing those in lab disease trials, or for looking at prevalence in the wild. So we can either model it or we can calculate it based on probabilities.
so you just want an R0 greater than 1?
Yep.
And that's good enough to make it?
I think it's good enough to keep moving forward. But I mean, I don't have like a bunch of R0s like memorized. But I know that like mumps, for instance, measles, for instance, is like an R0 of 3. So that's like a little bit higher. It's pretty good at transmitting itself. It doesn't have anything to do with mortality. It's just how many more infections is it going to make?
So you're not considering mortality rate in that?
Yeah. So we can kind of look at that separately. Back.
So one of the groups of people that you acknowledged at the end of your presentation are commercial fishers. I'm wondering how they feel about this kind of research, especially if you're talking about populations who are further downstream, considering we're a headwater state. What your reaction is to that?
Yeah, so I mean, it is kind of a tricky thing because there's like different interests involved in this. I don't know. I'm not familiar with sort of like how commercial fishing is done further south from here. But in this state, I mean, even part of like being permitted to commercially fish a lake is that you're agreeing to remove carp from that lake. Okay. So it's something that's like I can see the disjoint with it, but I can also see how these things could actually go together. So yeah, I mean, there's some conflicts, but there are also some common ground there. Yeah.
I wonder if you can go back to the histogram that shows percent of infected fish across different lakes.
This one.
No, not this one. The one where the
this
yeah,
one.
Yeah,
Here we go.
exactly.
Yeah.
So, can you explain a little bit more where the samples were collected from? Was it like a bunch of lakes?
So it's actually from one really big lake at different sites in the lake. It's Lake Biwai in Japan. So it's like a gigantic lake. and basically there was an outbreak event. I can't remember what year, but a couple years later they went back in and did sampling from a bunch of different sites along the lake.
And they were finding, because the smallest group could be kind of like 8-0. An interesting thing is that they start spawning at around 37 years.
Around here.
Or the next one.
Or the next one.
Yeah.
And so you see like, some.
So would that suggest that they just didn't catch it because they weren't alive yet? I think it was a couple of years before.
So they're actually catching these fish, but they don't have any KHV.
I mean, the fish didn't catch the virus because they weren't alive when it was around.
So they're in the same population. So they're finding them kind of like in the main lake. So they are. I mean, it's possible that like the smaller fish are hanging out with the bigger fish, and so they're not catching it yet. But they're sort of all in this same population at this point. But yeah, that's kind of the thought is they haven't caught it yet. And that if they do catch it, maybe the reason why they don't show up on this histogram is because they died instead of surviving and going into this gray category.
Yeah, I was just wondering, because those are like immature versus mature, not this. Why would a 25 centimeter fish be 0 out of 16?
Yeah. So you're like interested in like the biological kind of reason for that? I don't think that anyone's looked into that yet. I do know that it's something that I'm learning about right now, but I know that there are some differences in the development of the adaptive immune system in fish. So it could be that like at these 8 time points, like maybe that adaptive immune system is kind of kicking in. But I don't know for sure. That's total speculation that I'm curious about.
So you talked about sort of the temperature that the virus becomes most virulent. If that's the way to describe it, I'm not huge into microbiome stuff. But I was curious, has there been any work with sort of looking at virulence against just water quality in general, different parameters, conductance, phosphorus loading, turbidity, things like that?
So there is it's not really a great answer
like
to your
stress
question.
levels or something on the fish.
Yeah. So it's not a great answer to your question because I don't know of any that are associated with shedding. But just like presence of the virus in the environment, usually places that are more turbid, there'll be more of it, and if there's more like kind of like structure like plants and stuff like that. And I think the reason why that might be is just because the fish shed it and then it's able to hang out in the environment. But if bacteria are there, kind of like gobbling up the virus, then it'll be a little more ephemeral. But yeah, I can't think of any. Do you want to weigh in on that, Nick?
Light sensitivity.
Oh, light sensitivity is another thing too. Yeah.
How does it work?
Actually, this is an interesting thing I just learned about. So there was a study where they were looking at whether nets that had been exposed to KHV could transmit the virus to other fish. So they basically just get a bunch of fish in a net that are infected, and then take the net out and put it into another tank with naive fish, and there wasn't any transmission. But then Keith Way recently did a study where he did the same thing, but he put the nets in a dark place first, and like he didn't expose them to light. And then when he tried that again, he was able to transmit the virus using light in the net. So if it's dark and cold and damp, like you might, or cool and damp, then you might be able to keep it alive.
So this might be a misconception on my part, but isn't an environment that's turbid, has a lot of structure, an environment that Carp like to be anyway?
I don't know, maybe Epidemics is a better answer to that question. But from what I understand, Carp don't necessarily like to be in those places. It's just kind of like they're associated with lakes that they kind of chicken or the egg, like what is it? Do they change the environment and then they do better in it, or do they? Yeah.
Okay.
But I don't know. Epidemics, maybe you wanna.
Yeah, I don't know what they like or dislike, but often the way things happen is you put few of them in a lake that is abundant and the lake will flip into the turbid state, the carp stay abundant.
Yeah.
Well, we looked across hundreds of lakes in our data, look at the presence of H0 young of the year. And overall, I think about 5% of the lakes had young of the year.
Yeah, so not very
The
many.
strongest predictor was actually water turbidity. The higher the more likely you would find young of the year.
Interesting.
I don't know. I think this is related to larval food abundance.
Anything else? All right, well, thank you again.
Just around 3 years ago. 3 years. So
Tracy, or did she want
I know, I was just asking you questions.
want me to walk with you and hang out? You want me to hang out and walk the camera back with you?
It's up to you. It's up to you. I think that when you ran really much, that's also, yeah, there's a lot going on, especially in the
because one of the areas saying that they don't see positive symptom on the young fish, is it because they all died, the infected ones, or is it because
they can't pathogen?
right, exactly. But they're not present in the lake, they have been present for at least 2 years. Or at least a year.